Hello everyone, welcome to my channel Anupma Biology Classes. In this video, we will see Kingdom Monera and this is the second part of Kingdom Monera. In this video, we will focus on Actinomycetes, Mycoplasma, Cyanobacteria, Archaebacteria, etc. This video is taken from CBSE Class 11th, Part 4th of Chapter 2nd. Now, let's start the topic. So, the first thing is actinomycetes. Before this, in the previous video, we discussed on bacteria. So now, what are the general characters of actinomycetes? Actinomycetes is a group of unicellular branched filamentous bacteria which resemble fungal mycelia. They grow in the form of radiating colonies in cultures and therefore commonly called ray fungi. So here in this picture you can see the ray fungi. They occur most commonly and abundantly in soil, fresh water, manure, food products and other substrata which is rich in dead organic matter. The filaments are septate, that means non-septate, branched and very thin like 0.2 to 1.2 micrometer in width, but sometimes break off into rod-shaped structures. They are gram-positive, chemoorganotrophic, saprotrophic bacteria which constitute an important component of decomposers. A few species are, however, pathogenic in human beings and other animals. Pathogenic species causes disease, especially skin disease. Most of the species are facultative and aerobic. A few species are motile. Asexual reproduction occurs by fragmentation. Here you can see conidia formation or oidia formation. And the endospores are not found in actinomycetes. Many species of actinomycetes produce important antibiotics such as streptomycin. So this is the general character of actinomycetes. Now the second one is which is Mycoplasma. It is also from the kingdom Monera. So what are the general characters of Mycoplasmas? In 1898, the French scientists E. Nocard and E. R. Roux discovered some peculiar organism from pleural fluids of cattle suffering from bovine pleuropneumonia. Similar organisms were later isolated from other animals and also from human beings. These are named as pleuropneumonia-like organisms. It means PPLO. Borel et al. In 1910 named these organisms as Asterococcus mycoides. These organisms were later put under the generic name Mycoplasma by Novak in 1929. In 1966, International Committee of Nomenclature of Bacteria placed Mycoplasma under the class Mollicutes, which consist of two genera, Mycoplasma and Achloplasma. Mycoplasmas are the typical monerans, that means prokaryote, that occur saprotrophically in soil, sewage water and in decaying organic matter. They can survive without oxygen. Some mycoplasmas occur parasitically and pathogenetically in plants, animals and in human beings. Pathogenic mycoplasma causes disease. Mycoplasma are unicellular, non-motile, gram-negative bacteria, that means prokaryotes, which lack a distinct cell wall and do not produce spores. So, before this actinoplasma, that means actinomycetes, are gram-positive and these mycoplasmas are gram-negative. The cell membrane is made up of protein and lipids. It also contains cholesterol. The outer cell membrane encloses the dense cytoplasm which contains numerous ribosomes, and you know that ribosomes in case of prokaryote is 70S type, a network of fibrillar DNA, soluble RNA and soluble proteins. The DNA of mycoplasma are large enough to coat only for about 600 to 1000 cistrons per cell. Cells of mycoplasma divide unevenly into very minute bodies called minimal reproductive units. They are commonly formed inside the mature cells. These can pass through bacteria retaining filters. So, mycoplasma are the smallest free living microorganism. So, what is the size of mycoplasma? Mycoplasma vary in size mostly from 0.125 to 0.150 micron in diameter. They are pleomorphic. Pleo means many and morphe means forms. So, they are present in different forms varying in shape from spherical to branched filamentous structure. Since the organism have no definite shape, they are called jokers of the plant kingdom. They are unicellular, non-motile, gram-negative prokaryotes which lack a distinct cell wall and do not produce spores. Cell membrane of mycoplasma is made up of proteins and lipids. It also contains cholesterol. And cells of mycoplasma divide unevenly into very minute bodies called minimal reproductive units. So this is a short knowledge, short characters of the mycoplasma. Now, mycoplasma are resistant to the wall attacking antibiotics such as penicillins. Their growth is inhibited by tetracycline and similar antibiotics that act on metabolic pathways. Some common genera include in the group that is mycoplasma are, and these genera are, first one that is the mycoplasma which you know, second is urea plasma which you can see in this picture. This green structure is known as urea plasma. Then here you can see in this picture E. coli plasma, aneroplasma and spiroplasma. So these are the some genera of the mycoplasma. Now, next one is Cyanobacteria, which is also known as blue-green algae. So, what are the general characters of the Cyanobacteria? At first, see the occurrence of the Cyanobacteria, where Cyanobacteria are found. So, Cyanobacteria in Greek, you know that cyano means blue and bact means rod. So, these are blue rod-shaped structures. So, it's also known as blue-green algae. Are one of the most successful groups of prokaryote, gram-negative, photoautotropic organisms distributed all over the world. They have survived most successfully for more than 3 billion years on Earth. Cyanobacteria occupy a wide range of habitats and grow almost everywhere wherever they get suitable moisture and temperature conditions. They are predominantly aquatic and occur mostly in the freshwater habitats such as ponds, pools, lakes, and other freshwater reservoirs. Many species are terrestrial and grow on moist rocks, moist soil in cultivated and uncultivated lands and tree trunks. A few species are marine and form a brackish zone on the rocks at high tide mark. They occur as phytoplanktons, blooms, epiphytes, or as benthic formation on mud. A few species occur as endophytes or endozoic. Some cyanobacteria grows in most unusual habitats. A few species grow in hot water spring having a temperature range of 70 to 75 degrees centigrade like Formidium, Mastigocladus, etc. and other grow at very low temperature in the polar regions like Nostoc, Schizothrix, Microcoilus, etc. So, the red color of the Red Sea is due to abundance of the planktonic cyanobacteria, and the name of this cyanobacteria is Trichodesmium erythraeum. Some cyanobacteria live in symbiotic association with the other organism, and for example as algal partner in case of lichens, in Cycas corylide root, in water fern azolla, in Anthoceros thallus, which is a bryophyte, etc. Now let's see the organization of thallus. The organization of thallus ranges from unicellular to branched heterotrichous forms. So, first here see here, this is the unicellular forms. An example is Corococcus. Unicellular polar thalli, which with a definite base and apex, found in Chemisiphon, here you can see in this picture, and Dermocarpa. Multicellular colonial forms like, see here, this is Glyocapsa, Trichodesmium, Merismopedia, and Microcystis. Simple branched filamentous form without heterocyst and echinates, like in this picture you can see this is Oscillatoria. Next is Anthrospira, then Spirulina, and after this some are also unbranched like Formidium, Ligbia, Symploca, Microcellus, Cygothrix, etc. Simple unbranched filaments with heterocyst are found like in case of nostoc, Anabina, Allosoria, etc. Unbranched heterocystus filamentous with base and apex are found in case of Rivularia, Gloetrica, etc. Heterotrichous filaments with false branching like Cytonema, Plectonema, Tolipothrix. In this picture you can see the Tolipothrix, etc. So these are heterotrichous filaments with false branching. Heterotrichous filaments with true branching are found in Fissurella. In this picture you can see, and Haplosiphon. Or the next one is Stegonema. So these are the heterotrichous filaments with true branching. Flagella are completely absent, but the movements occur in some genera by spatial gliding motion, and such movements are connected with the secretion of mucilus. The genus Oscillatoria exhibit pendulum-like oscillating movement of its anterior region. Now, let's see the cell structure of cyanobacteria. So, in case of cyanobacteria, this is the ultrastructure of the cyanobacterial cell, and each cell has a definite cell wall. Here you can see, usually surrounded by a mucilaginous sheath. The sheath may be thin or thick, haline or pigmented, homogeneous and stratified. The sheath is made up of reticulately arranged microfibrils with an amorphous matrix. The fibrils may be composed of pectic acid and mucopolysaccharide. The cell wall is firm and rigid. It is two-layered. The outer layer or outer membrane is convoluted and the inner layer is smooth. The inner layer is made up of peptidoglycans similar to the bacterial cell wall. The cell wall is followed by plasma membrane, which is made up of lipids and proteins. Inner contents of the cell may be distinguished into two regions. Outer pigmented region called chromatoplasm and the central hyaline region which is known as centroplasm. The membrane-bound structures like two mitochondria, chloroplast, endoplasmic reticulum, Golgi body, true vacuole, etc. are absent. The photosynthetic pigments are located in the broad sheet-like lamellae which is known as thylakoid. The thylakoids are restricted to the peripheral region of the cytoplasm, usually arranged in two or more parallel stacks. Some lipid globules occur within the thylakoids, whereas phycobilisome particles are attached to their surface. Some authors suggest that these lamella also provide the sites for cellular respiration. The photosynthetic pigments present in the cells are chlorophyll A, beta carotene, myxoxanthophyll, myxoxanthin, C-phycocyanin, and C-phycoerythrin. The central nucleoid, see here, this is the nucleoid region, which is known as circular DNA, have several irregularly arranged fine strands of DNA, and these DNA strands are naked, that is, they are not associated with the histone proteins. In this respect, they resemble the circular chromosome of bacteria. The nucleolus is absent and the nucleoid is not bounded by a nuclear membrane. Other cellular inclusions like the gas vacuoles, cyanophysin granules, volutin granules, beta granules, polyhedral bodies, 70S ribosomes, etc. are present in this. The gas vacuoles are common in planktonic forms. They are the vesicle filled with the gas and bonded by the single membrane. They serve to regulate the buoyancy of the planktonic form in diffuse or low light intensities. They are large and help to bring the cyanobacteria at the surface. Some cyanobacteria possesses a special type of cells called heterocyst. Here you can see the structure of the heterocyst, which you can see, which is sky blue in color. To perform special functions, like this is the structure of a nostoc, anabena, and cytonema. Heterocysts are usually large-sized, rounded, and thick-walled cells devoid of nuclei. They are the sites of nitrogen fixation. There is no mitotic cell division in cyanobacteria. The cells divide by the fission or amitotic division. So this is all about the cell structure of the cyanobacteria. Now, let's see the nutrition in cyanobacteria. Cyanobacteria contain chlorophyll A and other photosynthetic pigments. They synthesize their own carbohydrate food from carbon dioxide and water in presence of light by the process called oxygenic photosynthesis. Now, reproduction in cyanobacteria. Cyanobacteria mostly reproduce by vegetative and asexual methods. Vegetative reproduction occurs by fission, fragmentation, and by the formation of harmogonia. Unicellular forms multiply by binary fission. Filamentous forms reproduce by breaking of filaments into short pieces. Asexual reproduction occurs in case of cyanobacteria by the formation of echinates. Here you can see this is the process of asexual reproduction where thick-walled perinating spores with stored food material. Next is endospores, that is, spores produced endogenously within a vegetative cell. Next is exospores, that is, spores produced exogenously. So, difference in endospore and exospore is endospore present within the cell and exospore is present outside the cell. And then nanocytes. So what are nanocytes? That is small naked spores produced inside the parent cell. So this is the method of asexual reproduction in cyanobacteria. Now there is no sexual reproduction, but genetic recombination equivalent to sexual reproduction has been reported in some genera like Cylindrospermum. In this picture you can see Cylindrospermum majus. Majus is the species name. Anacystis, redulans, and anabina species. All the three kinds of bacteria, parasexuality, that is conjugation, transformation, and transduction, have been reported in cyanobacteria. So this is about the reproduction. Now, what are the economic importance of cyanobacteria? So, in case of economic importance, there are some useful activities and some harmful activities are also present by cyanobacteria. So, what are the useful activities? Ability of cyanobacteria to fix atmospheric nitrogen, particularly in the wetland conditions as in rice fields, is highly beneficial phenomena in nature. This property is being explored as green manure by the farmer to enrich the soil with nitrogenous salt. Spirulina, a non-toxic fast-growing cyanobacteria, is being used as a source of protein-rich supplement to diet of human and also aquatic life. It is cultured, dried, powdered and then supplied in the form of 1 gram tablets. The process is known as single cell protein, which is known as SCP. Balls of nostoc commune are collected, boiled and consumed as food by the Chinese and South Americans. Their prepared food is called yo cho. Certain cyanobacteria like nostoc, cytonema, cylindrospermum, eulosoria are used in the recalmation of ooser land, that means sterile alkaline soil. These organisms secrete acidic chemicals which counteract the alkalinity of the ooser soil. Some cyanobacteria growing symbiotically in lichens as act like pioneer of plant succession on the barren rocks and help soil formation. Some cyanobacteria such as Anabina, Lingbia, Tolipothrix, etc. prevent soil erosion and help its conservation. Anabina and spirulina, produced in huge amount in Sambar Salt Lake in Rajasthan, is used as green manure by the local farmers. Some species of Anabina and Eulosoria do not allow mosquito larvae to grow in their vicinity. These species can be inoculated in ponds to prevent the growth of mosquitoes. Now, these are the useful activities. Now, what are the harmful activities of cyanobacteria? Some cyanobacteria like Mycocystis, Anabina, Rivularia, etc. release toxins in the surrounding water which is harmful to fishes and other aquatic fauna. Consumption of such water may be toxic to man and other animals. Some cyanobacteria grow on buildings and historical monuments in rainy season. They causes discoloration and severe damage to the plaster and brickwork. So here you can see this in this picture. These green colors are the cyanobacteria which are responsible for the damage of the building. Some forms such as Oscillatoria grow in municipal water reservoirs and water channels. They change the odor and spoil the taste of water. Some cyanobacteria causes damage by growing on textile, cordages and tents. Lingbia may cause some skin infections. Now, after this, see here the next one, that is the Archaebacteria. So, what are Archaebacteria? Archaebacteria are a unique type of prokaryotic microorganism living in most extreme of environments such as submarine, hydrothermal vents, the Great Salt Lake, the Dead Sea, the edge of the ocean, volcanic walls, hot sulfur springs, and other similar hostile conditions. Since these organisms continue to live under such conditions, perhaps representing the kind of habitats that were available when the earth first formed, they are believed to be early forms of life and hence they are called Archaebacteria. Archae means ancient and bact means rot. So these bacteria are the primitive, that means ancient bacteria. So what are the general characters of the Archaebacteria? Archaebacteria are characterized by the absence of peptidoglycan in their cell walls. In most of them, the wall is composed of non-cellulosic polysaccharide. And in other, it is made up of proteins. However, in some other, there is no cell wall. So, here the outer wall is known as the cell wall. And here, after this, the plasma membrane is also present. So the cell membrane lipids of Archaebacteria contain glycerol-like linked to branched hydrocarbon chains. The nucleotide sequence of archaebacterial RNA are quite different as compared to those of other organisms. Now, what are the groups of archaebacteria? So, based on diverse living conditions and metabolic activities, the archaebacteria can be divided into three representative groups, and these are METHANOGENS or methanogenic archaebacteria, halophiles or halophilic archaebacteria, thermoacidophiles or thermoacidophilic archaebacteria. So we will see each and every part of the archaebacteria's group in detail here. So first one, that is the methanogens or methanogenic archaebacteria. These archaebacteria have some characters like methanogens are the strictest anaerobes known. They live anaerobically in the swamp, marshes, and in the gut of animal. They live as symbionts in the rumen, which is rumen is the first part of the stomach of herbivorous animals. They chew their cud, example like cow, buffalo, goat, etc. These murins help in fermentation of cellulose. Methanogens have been grouped under four genera like Methanobacterium, Methanobacillus, Methanosarcina, and Methanococcus. They are chemosynthetically bacteria that produce methane, that is CH4, by the anaerobic decomposition of organic matter. Hydrogen oxidizing methanogens convert carbon dioxide and hydrogen into methane. Acetoclastic methanogens convert acetic acid into the methane and carbon dioxide. Methane is a popular pollutant that contributes to the greenhouse effect and the global warming. The fermentation of cattle dung by the methanogens is especially designed plants, that is gobar gas plant, not other provided cooking gas, that is fuel gas, but also leaves behind a slurry which can be used as an organic manure for growing crops. If methanogens are added to a mixture of decomposers and animal waste, a biogas comes out which can be used to run a generator that produces electricity. So this is all about the methanogens. Now, what are halophiles? These are anaerobic salt lovers which occurs in extreme saline environments such as salt lakes, salt pans, salt marshes, brines, that means salt solution, etc. Some halophiles occur in deep sea volcanic vents at 110 degrees centigrade, a temperature at which the water remains liquid because of extreme hydrostatic pressure. So, in strong light, the halophiles develop a pigmented membrane, that is purple membrane, composed of a pigment called bacteriorhodopsin, related to the one found in our own eyes also, to harness the sun's energy. They carry out a unique version of photosynthesis in which the carbon dioxide is not converted to sugar. Instead, the light energy is utilized to carry an ATP production by chemoosmosis. Examples of halophiles are, here you can see that, Halobacterium and Halococcus. Now, what are thermoacidophiles? These archaebacteria live where it is both hot and acidic, such as hot sulfur springs where temperature remains around 80 degrees centigrade and pH around 2, that is highly acidic. Under aerobic conditions, these organisms oxidize sulfur to sulfuric acid. Under anaerobic conditions, some thermoacidophiles reduce sulfur to hydrogen sulfide. Examples of thermoacidophiles are sulfobolus, here in this picture you can see, thermoplasma and thermoproteus. So, archaebacteria are the oldest of the living fossils. They represent the oldest form of life that has survived almost all the geological changes on Earth. They are the first organism who experimented with the absorption of solar radiation to get their energy, survived under extreme anaerobic condition, and they developed the novel method of oxidizing chemical substances with the availability of oxygen. So this is all about the Actinomycetes, Mycoplasma, Cyanobacteria, and Archaebacteria. So this is the completion of the Monerans, kingdom Monera. So in the next video we will start kingdom Protista. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.